Hello again. Good evening. Um, Welcome to study session 2. Here we'll be talking about the generation of ATP in glycolytic pathway. Okay, so we've already laid a foundation as to what glycolysis is and why the glycolytic pathway is important. I think I've presented this slide in the last study session. So basically, it's a central pathway, a central catabolic pathway, but it's essential in metabolism because some of its precursors go into the biosynthesis of macromolecule, while other precursors or other intermediates of this pathway um, find their way into the citric acid cycle and the electron transport chain, where they lead to the production of the energy currency of the cell, which is ATP. That said, let us go back to your lecture note. All right. So the basic introduction here is we already summarized from the last study session that glycolysis produces two ATP molecules. ATP is the energy currency of the cell. Okay. However, more ATP is produced when pyruvate is completely oxidized to CO2 and H2O. This is where we delve into the citric acid cycle. Okay. In the mitochondria. The electron transport chain, the glycolytic pathway is regulated through the activities of three enzymes, which I have also mentioned, those irreversible enzymes in the glycolytic pathway. These are the enzymes. You see, this is reversible, but this reaction is irreversible. It is catalyzed by hexokinase. The other irreversible reaction, yeah, 3, catalyzed by phosphofructokinase 1, and then the last one, catalyzed by pyruvate kinase. Okay. Yeah. So this is what has been passed here. This is the message that has been passed here. Okay, the glycolytic pathway is regulated through the activities of three enzymes that catalyze its irreversible reactions. Okay. However, the most important control element of glycolysis is the enzyme phosphofructokinase, PFK, which is the third enzyme, which catalyzes the third reaction of glycolysis over on this. Okay. All right. This 3, phosphofructokinase 1. So phosphofructokinase 1 is the most important regulatory enzyme in the glycolytic pathway. So the enzyme catalyzing the first irreversible step unique to the pathway. The pyruvate has 3 fates. It may be converted to acetyl-CoA, which we'll see, ethanol or lactate. Depending on the type of metabolism, it can be aerobic, and the organism, it can be aerobic organisms that take in oxygen. If the metabolism is happening in the presence of oxygen, that's aerobic. It's anaerobic in the absence of oxygen, and that's where you have anaerobic metabolism. So the clinical conditions associated with impaired glycolysis include lactic acidosis and pyruvate kinase deficiency. We'll dive into this in a minute. Okay, so going back this slide. So these reactions summarize the checkpoints in glycolysis. Those 3 reactions that can be regulated because of the enzymes. Now, this is the first glucose to glucose phosphate, hexokinase. All right. So this the reaction is irreversible. This enzyme is a regulatory point, but the most important regulatory enzyme that catalyzes the rate limiting step of glycolysis is phosphokinase. Okay, and the last regulatory checkpoint is the reaction catalyzed by pyruvate kinase. The last reaction of the glycolytic pathway, conversion of phosphoenol pyruvates to pyruvates. All right. So basically, this is it. Here we talk about the energy investments of ATP and then the payoff, the production of ATP in the latter stage of glycolysis, which I have already explained in my first study session recorded video. So here, how many ATP are produced in each glycolytic pathway? Of course, it's 4. But if you subtract the two ATP that were invested in the first preparatory or first stage of glycolysis, then you have a net total of 2. So in case we forget, let's present this again. So in the preparatory phase, we are investing 1 ATP here in this first reaction. And then in the third reaction, we invested another ATP. In the payoff phase, stage 2 and stage 3 of glycolysis, according to your lecture notes, ATP is produced here. You see the arrow going out, that's product, not substrate. It's not coming in, but it's going out. So here we've invested 2 ATPs. Here we have total 2, 1 here, 1 here. But here we have 2 ATP produced. We have another 2 ATP produced. So 4 ATPs are produced in glycolysis. But if you subtract the number of ATP invested from the total you have here, you have 4, you invested 2. So you have 2 remaining. So realistically, right, glycolysis yields 2 ATPs. Okay? So how many ATP are produced in each glycolysis pathway? 4, but I think it's really 2. Because if you go back to study session 1, there's a similar question, although not coined like this, but the answer was 2. So you could disagree with this, and you would be correct. So the regulation of glycolysis, right? Okay. The glycolytic pathway has a dual role, okay? It degrades glucose to generate ATP, and it provides a building block for synthetic reactions, which I think was reiterated in the study session 1. So the rate of conversion of glucose into pyruvate is regulated to meet these 2 major cellular needs. When you have enough ATP, then the glycolytic pathway needs to slow down. And when you don't have enough energy in the cell, the glycolytic pathway is going to be activated, okay? When there's a need to synthesize some macromolecules in the body, the anabolic reactions will become active. Basically, the rate of conversion of glucose into pyruvate is regulated by these 2 major cellular needs. And it decreases glucose to generate ATP, provides building blocks for the synthetic reactions. So these cellular needs, the need for energy and the need for biosynthesis of macromolecules in the cell, or molecules in the cell, not macro only, right? Will be the cellular needs that inform the regulation of glycolysis, conversion of glucose into pyruvate. So enzymes catalyzing essentially irreversible reactions are potential sites of control, okay? So these enzymes, 1, 3, 10, hexokinase, phosphofructokinase, pyruvate kinase. These are regulatory points. in the glycolytic pathway. So if I ask you a question in this regard, you should know what to tell me, right? So in glycolysis, the reactions catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase are virtually irreversible. So these enzymes play regulatory as well as catalytic roles, of course. However, PFK, phosphofructokinase, is the most important control element in glycolysis. The enzyme is inhibited by, now, of course, when you need energy in the cell, the glycolytic pathway comes into play. When you don't need that much energy, there should be a mechanism to regulate the glycolytic pathway. Now, one of those mechanisms is high levels of ATP, which is the product, right, of the glycolytic pathway, binds to a highly specific regulatory site that is distinct from the catalytic site. That's the allosteric site, which we've mentioned in the other study session in the first lecture. The inhibitory action is reversed by AMP, of course. AMP, adenosine monophosphate. ATP is the energy currency of the cell. AMP is not. So it reverses the inhibitory action of ATP. When you have low ATP in the cell, AMP will bind to the allosteric site of the PFK and to reverse the action of ATP. Basically, that's what we are saying. The activity of the enzyme increases when the ATP-AMP ratio is lowered, of course. Of course, this makes sense, doesn't it? High levels of citrates, the citric acid cycle, which we are going to dive into in a minute, or maybe in the next lecture, not this. So, but for you to understand this statement, let me show you the citric acid cycle, okay? You've been hearing about the citric acid cycle. So this is roughly what the citric acid cycle is. Now, this is a summarized glycolytic pathway. It's summarized here from glucose to pyruvates. Of course, you've seen the reactions in between. Now, pyruvates in aerobic respiration, the presence of oxygen is converted to acetyl-CoA, and this acetyl-CoA goes into the citric acid cycle. All right. Now, in the citric acid cycle, acetyl-CoA is condenses with oxaloacetate here to produce citrates. That's why this cycle is called citric acid cycle. Citrate here is citric acid, right? Now, this citric acid goes into a cascade of reactions to produce ATP and NADH, which goes into the electron transport chain in the mitochondria and generate more energy currency, ATP, and electrons as byproduct, CO2 and H2O, basically not electrons. I'm sorry about that. Okay. All right. So high levels of citrates, which indicates that biosynthetic precursors are abundant, will also inhibit the activity of this enzyme. When you have had enough production, bioconversion of glucose into pyruvate, and it has gone into the citric acid cycle, it is expected that you're going to have high levels of ATP and high levels of citrate, right? Citric acid, because there's high pace glycolysis going on. So of course they should inhibit the glycolytic pathway at some points, right? Then when the ATP or citrate concentration is low, then there's probably a need for glycolytic pathway because energy is low in the cell. So I hope this makes sense. Citrate inhibits PFK by enhancing the inhibitory effect of ATP. Okay, this is how citrate inhibits PFK. By enhancing the inhibitory effects of ATP, hexokinase and pyruvate kinase also participate in regulating the role of glycolysis. Okay, so in general, the enzyme catalyzing the committed step, that's the first irreversible reaction unique to the pathway in a metabolic sequence, is the most important control element in the pathway. PFK is most active when the cell needs both energy and building blocks. It is moderately active when either energy or carbon skeleton is needed, while the enzyme is almost switched off when both are abundant. Of course, this statement makes sense. There's a need for energy. Of course, PFK will be active because the glycolytic pathway breaks down glucose to produce ATP and reduced coenzymes NADH, FADH2, which go into the electron transport chain to give you energy. So when there's an energy deficit, PFK should be most active. Or when there's energy, relatively depending on the level of energy presence, right? It can be moderately active. And then when the cell is energy rich, okay, then there's no need for PFK to be active. So it should be switched off. So I think the statement is self-explanatory. All right. Okay. So I think basically this study session and study session one are not so different from each other. Okay. Hexokinase and pyruvate kinase also participate in regulating the rate of glycolysis. Pyruvate kinase from muscle and liver is allosterically inhibited by ATP. So the conversion of KEP to pyruvate is blocked. Of course, you know pyruvate kinase, you know hexokinase, you know phosphofructokinase. Hexokinase, on the other hand, is allosterically inhibited by glucose 6-phosphate. It makes sense, doesn't it? I mean, hexokinase catalyzes the conversion of glucose to glucose 6-phosphate. That's hexokinase. So the product here allosterically binds to an allosteric site in hexokinase. That's a site that's not the active site. And binding of glucose 6-phosphate, when enough glucose 6-phosphate is produced, and there's no need for the glycolytic pathway to continue, this glucose 6-phosphate will bind to that allosteric or regulatory site on the enzyme to prevent it from further converting glucose into glucose 6-phosphate. Basically, that's what we're saying. Okay. So this is just to prove the regulatory roles of these three enzymes. While pyruvate kinase from muscle and liver are allosterically inhibited by ATP. I hope you understand this concept of allosteric regulation. I've explained it in the previous lecture as well, but let me just revisit it. You know, sometimes we tend to forget things. This is the enzyme, right? Sorry. This is your enzyme. This is the active site, the catalytic site of your enzyme, where the glucose, the substrate, will bind to this site to produce an enzyme substrate complex, which will be now converted to the product. The enzyme is not used up in the reaction. Now, this enzyme has another site here. This now is the regulatory site, the allosteric site, where the product inhibits the enzyme from further converting your substrate into products. So for example, if say this is pyruvate kinase, ATP will bind to this regulatory site to prevent the conversion of phosphoenol pyruvate to pyruvate because we've had enough ATP already in the cell. You get it? So basically that's what we are seeing. In the case of hexokinase, for example, it converts glucose to glucose 6-phosphate. So enough glucose 6-phosphate, if enough glucose 6-phosphate is produced and there's no further need for energy, the glycolytic pathway has to slow down or be temporarily halted. Glucose 6-phosphate to prevent hexokinase from further converting glucose to glucose 6-phosphate. That glucose 6-phosphate will bind to the regulatory site, the allosteric site, this site of hexokinase, and binding of the product to this side will prevent further conversions. So this is a way of regulating the activity of this enzyme and other enzymes as well. Allosteric sites. These are very important terminologies in allosteric site, active site, and things like that. These are very important terminologies in biochemistry. Okay, so um, okay, so metabolism in the absence of oxygen. In the presence of oxygen, you have your metabolic flux going from pyruvate to acetyl-CoA and to the citric acid cycle, then to the electron transport chain. However, in the absence of oxygen, for example, athletes who run, the runners, the athletes, the relay runners, and people like that who do high intensive exercises, sometimes they run short of oxygen. Their muscle cells convert in their glycolytic pathway instead of the pyruvate to go into the mitochondria acetyl-CoA citric acid cycle electron transport chain. That doesn't happen. The pyruvate goes into an alternative path where it's converted to lactate, which this lactate accumulates and causes lactic acidosis. I think one of those symptoms is muscle pull. People who play football or people who run will probably tell you about this. As nurses, since you're a nursing student, you should know about this muscle pull. When suddenly an athlete ceases to feel the legs, maybe they can't move the leg or something like that because there's high acid accumulation in the muscles, lactic acidosis. All right. High lactic accumulation, sorry, in the, yes. So this is because of this reaction that happens here, pyruvate. There's no enough oxygen to facilitate the conversion of pyruvate to acetyl-CoA. So pyruvate is converted to lactate. And this reaction requires the reduced form of this coenzyme NADH, which is oxidized to NAD plus, while pyruvate is reduced to lactate. Okay, so this reaction is one of two reactions that are called anaerobic fermentation. The other reaction, which occurs in mostly microorganisms, is the conversion of pyruvate, of course, also in the absence of, not absence really, let's say in the absence of oxygen, pyruvate gets converted to acetaldehyde and then to ethanol. Now, this would, well, we are seeing it here from a human perspective, the glycolytic pathway, because the glycolytic pathway in microorganisms can be a little bit different. Some reactions are not the same as in humans. We know some yeasts are encouraged scientifically, of course. They are in some inductions, some substrates are added to induce production of ethanol. Say if your target is producing bioethanol or things like that. But that's not the focus of this course, so we're not going to go into that. But in the absence of oxygen, like you see here, pyruvate can be converted into acetaldehyde, and then acetaldehyde is converted into ethanol. Okay, so let's keep it on track. So these are the reactions which you've also seen in the slide. So you can take your time and read the lecture note with an open mind. Biochemistry is very easy to understand if you are open-minded and you don't give up very easily. So, okay, so a lot of energy is derived aerobically by means of TCA. Now, the question would be, which is better, right? Which produces more energy? Is it the aerobic glycolytic pathway or the anaerobic glycolytic pathway? Aerobic as in the presence of oxygen, or anaerobic, as in the presence or in the absence of oxygen. Of course, the aerobic pathway yields higher number of energy currency than the anaerobic pathway. Okay, because looking at this pathway, it's even more extensive. Your reduced coenzyme go into the electron transport chain, NADH produced from both the glycolytic pathway and the citric acid cycle will go into the electron transport chain and produce ATP. I told you earlier that one NADH produces 2.5 molecules of ATP, while one FADH2 produces 1.5 molecules of ATP. Okay, so this is your Krebs cycle. So this is probably not obtainable in the anaerobic fermentation. That's both the lactic acid through the lactic acid mechanism or through the ethanol mechanism. Okay. Now, between the conversion of pyruvates to acetyl-CoA is catalyzed by an enzyme called this reaction, pyruvate to acetyl-CoA here, because acetyl-CoA is what enters the citric acid cycle. Now, pyruvate to acetyl-CoA is catalyzed by an enzyme called pyruvate dehydrogenase complex. This enzyme is a complex, not just one enzyme, but three enzymes working together. So I think it should fit the description of what we call a trifunctional enzyme. This enzyme requires NAD plus as a coenzyme, which is reduced to NADH, which also goes into the electron transport chain, right? CoASH as an input, which produces the acetyl-CoA here. And then see, it gives off CO2. It requires coenzyme FAD as well. You have TPP, thiamine pyrophosphate, which is needed by this enzyme complex, and lipids. Lipoic lipids is also required by this enzyme. So this is very important. Well, say we ask you to list the coenzymes required by pyruvate dehydrogenase complex, you should be able to tell us, right? Okay. FAD, lipid, TPP, NAD plus, No, not really. NAD plus, it's part of the reaction, really. But the coenzymes here are TPP, FAD, and lipids. Okay, so look at the Krebs cycle. It's very, very beautiful cycle. Very beautiful. Okay. So acetyl-CoA is condenses with oxaloacetate here to produce citric acid. Citric acid is converted to, there's the enzyme that does this. In the next lecture, which is study session 3, which is dedicated to citric acid cycle, we'll go into details there. We'll mention the enzymes and what they do. But here, summarily, citric acid is converted to isocitric acid, that's isocitrates. And in biochemistry, we use citrates, isocitrates, not acid. I'll tell you why also in the next class. Okay, and then isocitrate is isocitrate dehydrogenase decarboxylates. It removes a molecule of CO2 from isocitrates to produce alpha ketoglutarate. And this enzyme uses NAD plus, reduces NAD plus to NADH while oxidizing isocitrate to alpha ketoglutarate. Alpha ketoglutarate dehydrogenase decarboxylates alpha ketoglutarate to produce succinyl-CoA. Here we also have a reduced NADH produced just like this. All these coenzymes, reduced coenzymes, go into the electron transport chain to give off ATP. Okay. So succinyl-CoA to succinate produces ATP. This is called substrate level phosphorylation. You have production of ATP here in the citric acid cycle. This is the other coenzyme I kept mentioning in the previous class, and I think in the earlier part of this class, the early part of this class, FADH2. I'm going to ask you to go and find out the full name, the full meaning of FADH2 and NADH here. what's the full name, right? I can't tell you everything. So I think I will make this an official quiz or something. It will be part of my quiz. You need to find this out. Succinate is now converted to fumarate, fumarate to malate, malate, malate to oxaloacetate. NADH is also produced here. You know, this NADH, NADH, FADH2, NADH2, these are very, very important molecules. They are energy currencies by proxy. You know, they go into the electron transport chain and they produce ATP, just like I've mentioned. All right. So we'll go into these details in the next class, not here. Okay. So I've talked about this. Now, clinical symptoms associated with impaired glycolysis, of course, lactic acidosis. I've explained this already. This is most frequent form of metabolic acidosis because it occurs as a result of overproduction of lactate or underutilization of lactate or inhibition of pyruvate dehydrogenase. Okay, so if you inhibit this enzyme, you can there won't be production of acetyl-CoA and there won't be citric acid cycle. Instead, instead of production of acetyl-CoA, pyruvate will now be converted to lactate. So basically, inhibiting pyruvate dehydrogenase complex leads to anaerobic fermentation, right? So it makes sense. Okay. Inhibition of pyruvate dehydrogenase. So it may also be as a result of rare congenital disorders where mitochondria do not function at full capacity, or diabetic ketoacidosis, as well as liver and kidney disease. I think in the latter part of this course, we're going to discuss diabetes. So we'll key into this a little bit better then. All right, it is characterized by lactate levels greater than 5 millimole per liter and serum pH of less than 7.35. I think the biological pH is 7.4, give or take. You should know better than me, right? You are practicing nurses. So, I mean, I shouldn't see much here. All right, lactic acidosis, which you know, pyruvate kinase deficiency. A rare genetic defect of glycolysis causes hemolytic anemia. Glycolytic intermediates close to the pyruvate kinase step accumulates, whereas pyruvate and lactate concentrations decrease. Lysis of red blood cells may cause jaundice and increase bilirubin. So what do you really understand by this? If there's pyruvate kinase deficiency, where is pyruvate kinase? Let's make sense of these statements. So this is pyruvate kinase, enzyme 10 of glycolysis. If there's a deficiency of this enzyme, phosphoenol pyruvate and this intermediate here, 2-phosphoglycerate, 3-phosphoglycerate, will not reach the pyruvate step. There won't be conversion of whatever reaches here to pyruvate. So there will be accumulation of these intermediates, right? And looking at these intermediates, really, phosphoenol pyruvate, pyruvic acid, 2-phosphoglyceric acid, if we want to put things in context. So you have accumulation of compounds that are mildly, maybe not strongly, but mildly acidic, but the accumulation is not very good for the system, is it? Right? It's not. So as these compounds accumulate, of course, the endpoint will probably be the lysis of your red blood cells, jaundice, and other complications, you understand, which is not very good for the whole cellular health and things like that. So I think it makes sense, right? So here's an ITQ question. The most important control element in glycolysis is pyruvate kinase. So you can answer that and give us a reason. Okay, so 2 marked assignments. I think we'll address this. If I deem it's worthy to give you these assignments, I'll communicate in the DLC platform as usual. So I think I've done justice to this. Look at this self-assessment. I strongly advise you to go and attempt this self-assessment. It's going to be good for you, right? And I think I'll even give you this as an individual assignment. You should, try to answer these questions. If you study very well, these questions are very easy questions to answer. And then in the study session 3, which hopefully we'll be doing next week, we're going to dive into the TCA cycle, the citric acid cycle. People also call it the tricarboxylic acid cycle, but I hear there's a school of thought that frown against this tricarboxylic acid cycle because citric acid is not the only tricarboxylic acid. So using tricarboxylic acid cycle widens the scope. You wouldn't know what we are talking about because, of course, you understand, right? Anyway, let me not bore you with some of the scientific arguments. See you in the next study session, and thank you for your time.